Hello students, welcome to RJR Academy. This is biology session. In this session, we'll discuss about the plant physiology. In the last episodes, we discussed about human physiology. Next to human physiology, the weightage that we can give for the NEET preparation is the plant physiology. Here it comprises of 18 to 22% of the marks, the total you can expect from this particular unit, right? So I hope you're practicing all the test papers, and this session will be beneficial for you. Like if you have any queries or any doubts that if you have, and this will be like great platform for you, so that we can interact with each other. And if you have any doubts even with the session, you place your comments in a comments box, so that it will be helpful for us to clarify your doubts. All right, so let us begin with the session. The first question, 91. The cell derived from the meristems differentiate and regain the capacity to divide by. So this is a common thing that we discussed like earlier part itself in the regular class itself. Differentiation, redifferentiation, dedifferentiation. So I have correlated this part with the cell cycle also. So what happened during cell cycle? That we have the interphase and the mitosis part. So for your information, so I'm just giving a recap on this part. So when you look at the cell cycle, we have a G1 phase here, G1 phase, we have S phase, and we have a G2 phase. And this, for example, we take it as a M phase, prophase, metaphase, anaphase, telophase, and everything. And that will be in between, we have a small gap. This we call it as a GO phase. GO phase, or this we call it as a quiescent stage. Okay, I hope you can recollect with this one. So what happened, like there are certain cells which will be going for this GO phase for the temporary basis, like this will be better metabolically, the cells are active, but for short span of time this will go with this GO phase. And later part what will happen, the cells will be differentiated and form like some other structures, for example parenchyma cells, and this parenchyma cells later part it will be changed to the core cambium, and it will be specialized in function. I hope you can recollect this. We already discussed the process like differentiation, redifferentiation, dedifferentiation. So having this in your content, if you read this question, the cell derived from meristems differentiate and regain the capacity to divide by a phenomenon, it's a dedifferentiation. Dedifferentiation is like once it reached the differentiation and redifferentiation, again what will happen, the cells again come back to its own, the normal cell cycle. This we call it as a dedifferentiation. So what is this? The process of the despecialization of a differentiated cell, so that they become undifferentiated. Once it reach, for example, core cambium, after that like it cannot be differentiated again. It is specialized in function and able to divide, and this usually this process called as dedifferentiation. So we go with the option 2 for this question. Question number 92. Oxynometer is used for the measurement. This is very direct question. We usually use this oxynometer for the plant growth. So option 4 is the correct answer. Question number 93. The Avena curvature is used for the bioassay. This we done with the coleoptile test Avena. This was proposed by a scientist named Avena et al., where he placed, actually this is a coleoptile region, for example, you take. So what the Avena curvature test, which means a bioassay, which means like he placed gelatin, the agar blocks on the tip of the coleoptile. After like periodical observation, he started observing like the coleoptile will start bending away from the light, actually where the gelatin has been placed actually. So this bioassay which was performed to detect the conformation or the function of IAA, that is indole acetic acid. So because I told you, if you go to the video that the discussion, I mean the class part, the live class part that we had, if you have gone just recap and see, like we discussed for all the hormones, I mentioned the bioassay. If you would have seen like the IAA, the bioassay, the test that we use is a Avena curvature test. Why it's called as Avena curvature test? This is the coleoptile. This coleoptile which turns towards away from the light part where the agar block has been placed. This clearly confirms like the coleoptile, the tip of the coleoptile region where the indole acetic acid, the function which has been confirmed, and this bioassay which helps to determine the function of indole acetic acid. So for this particular question, the correct option is IAA, that is indole acetic acid. Okay, and simultaneously when go with the Avena, this was even confirmed by the scientist, we cannot forget the scientist Wendt. The scientist name Wendt in the year 1928. This can measure the auxins, which can measure the auxins up to 300 microgram per liter. So the quantitatively like 300 microgram per liter, this Avena curvature test can be measured. is a universal natural auxin and Avena curvature test, which acts like accurate. This is a very accurate bioassay for the plant hormone. So for this question 93, option 1 is the correct answer. Question number 94, assertion and reason. If you see, plant growth regulators, we call it as PGRs, are very important for plant growth and development. Reason, auxins do not induce flowering in gymnosperms. If you look at assertion and reason, both assertion and reason are true, but the reason is not the correct explanation for the assertion. If you see the justification, because if you see the plant growth regulators or the small simple molecules and diverse chemical compositions that you know, usually which present in what? Which present in the very low concentration as far as the growth hormones are concerned. What happens, like this may be one type of like plant growth hormones of the or the phytohormones you take. Auxins are the phytohormones, they do not induce the flowering in a gymnosperm. So for example, gymnosperms with the naked seeds, where you could see the same hormones that you could not see in case of gymnosperms, only in the angiosperm this can be functional. So having this is a content, right, where you could see assertion and reason, both assertion and reason are correct, but the reason is not having any correlation or any link with the assertion. So we go with the second option for this particular question. Question number 95, the hormone used in a suspension culture. Suspension culture, if you go, out of the 4 option, we go with 2,4-D. So 2,4-D, again the derivative of like indolactic acid, the synthetic. So we have the natural auxins and the synthetic parts that we would have been classified, right? So the hormones usually we used for the suspension cultures in the artificial condition is a 2,4-D. Question number 95, 2,4-D is an artificial auxins where we can go for the suspension culture. So whenever the suspension cultures, you go with 2,4-D. Question number 96, which one of the following is plant functions is not controlled by auxins? As we discussed already about significance on uses of every hormones, the plant growth regulators. Looking at this, the first hormone, the auxins, apical dominance, phototropism, growth, which all the main functions which related with what? Related with the auxin, but photosynthesis. Photosynthesis is odd man out, where auxins control the apical dominance and other functions, but it's not related with photosynthesis. So we go with 4th option for this question. Question number 97. Compare the statements A and B. Statement A, auxins promote apical dominance by suppressing the activity of the lateral buds. Statement B, in moriculture, periodic pruning of shoot tips is done to make mulberry plants bushy. So having this 2 statements, if you correlate, right, if the correct options go with both the statement A and B are correct, and A is the reason for a B. So if we go with the option 3 for this particular question, that is 97. Question number 98, the potent weed killer. Weed killer, usually for the weed, will be the great nuisance for the farmers and the people who are involved in agricultural perspective. The potent weed killer often we use is a 2,4-D. It's a synthetic derivative. 2,4-D is commonly used. This is 95 as well as for 98. The same answer. 2,4-D. This seems to be synthetic hormones. Synthetic hormone. So 2,4-D is the correct answer for this question, that is 98. Question number 99. Gibberellins can promote seed germination because of the influence of, how it can influence or promote a seed germination, because influencing a production of what? Production of hydrolytic enzymes. When you see the hydrolytic enzymes, you would have studied the 2 types of like epigeal and hypogeal type of germination that you would have discussed. We discussed already in monocot plants and dicot plants, how the elongation of hypocotyl takes place, how this process takes place as per germination of dicots and the elongation of hypocotyl, where the seeds which sprouts out of the ground surface, but this won't happen in case of like monocot seeds, where you could see coleoptile and coleorhiza. What is the mechanism for this? Out of the 4 options you see, that is because of the production of hydrolytic enzyme. Which is responsible for producing hydrolytic enzymes? Gibberellins. Gibberellins is responsible, this particular hormone is responsible for producing hydrolytic enzyme. This hydrolytic enzyme enhance the elongation of hypocotyl. So for example, if you know the structure, right, hypocotyl, this if you could see the seed, this is epicotyl, and this one you could see hypocotyl. And what happens, this hydrolytic enzymes which makes the hypocotyl to elongate. So this hypocotyl elongates, and what happens, automatically the seeds will start sprouting towards the upper surface, automatically the seeds which will be coming out of the ground. So the main reason is because of the gibberellin, this hormone which produces the hydrolytic enzyme. This makes the elongation of hypocotyl, and this hypocotyl make the seeds to sprout out of the soil surface. Especially this you could see in case of dicot seeds and not with the monocot seed. The reason why not in a monocot seeds in maize grains you cannot see this is because where you could see the coleoptile and coleorhiza in case of monocot seeds or grains, not with the dicot seeds. So for this question 99, option 2 is the correct answer. Then question 100. Which of the following plant growth hormones increase the yield of sugar by increase the length of stem in a sugar cane? This you will study in the biotechnology part also, like Saccharum officinarum, barberry, like North Indian sugarcane, South Indian sugarcane, having that as a basic the concept that in your mind. But when you go detail, like which is responsible for increasing the sugar content of the sugar canes, this is because of the gibberellic acid. The gibberellic acid which plays a very important role to increase the why? Because it increases the internodal length in the plant's stem store sugars in the sugar cane. What happens, like the gibberellic acid which used to increase the sugar. It means that some sugar canes, like it is not up to the mark, even if you taste it, it's not giving good taste for you because the sugar content seems to be less. Why the sugar content? You cannot taste the sugar cane. This because the gibberellic acid which is responsible for especially the interlodal length, because interlodal length seems to be reduced, then sugar content also will be reduced, automatically the sugar cane will not be tasted. So the credit goes to gibberellic acid. For this question, option 4 is the correct answer. Question 101. Internodal elongation is associated with the same question that we discussed, one that is 100 and 101. Both are same type of question. You go with the GA, that is gibberellic acid is the correct answer. Either sugarcane content, internodal elongation, any type of question comes, you just go with the gibberellic acid, no doubt in this one. So we're done with question 100. Question 101. Internodal elongation is associated with gibberellin. Question 102. Genetically dwarf plants can be induced to grow tall by using, for example, the plants are very dwarf, actually it's not growing beyond its length. So what is actually the choice that we can do it? We can inject gibberellins so that the plants which can grow taller. So this is the choice by which the genetically dwarf plants, if it is confirmed that genetically the plants only grows only this much of size, it cannot grow beyond this particular level, then we can induce, we can mutate, and by which what happens, the genetical structure, the genetical sequence can be altered by means of introducing this gibberellins, and this can manipulate the sequence of the plant. So automatically genetically dwarf plants which can be increased in size. So for this question, option 1 is the correct answer, question number 102. Question 103. The plant growth regulator kinetin is an, what's a derivative? This also we discussed for every hormone. I hope you can recollect in the class we discussed purine derivative, adenine derivative, everything that we discussed, right? So having this in your mind, kinetin is a derivative. What's a derivative? It's an adenine derivative. Question number 103. 103, it's an adenine derivative. So if we talk about adenine, so why we call this as an adenine derivative? This is because is a first cytokinin to be discovered. This kinetin is the, always remember, kinetin is the first cytokinin to be discovered. Chemically, if you call it a cytokinin, is a 6-furfuryl aminopurine. This we call it a 6-furfuryl aminopurine. Maybe you can get this type of question also. Sometimes they'll give the cytokinin and they'll ask what is the chemical nature. So if they ask the chemical nature, then you should go with 6-furfuryl aminopurine will be the correct answer for this one. So what happens where the purine is adenine and kinetin does not occur naturally. So for this question, you go with the question number 103, option 1 is the correct answer. Question 104. Coconut milk stimulates cell division as it is a rich source of, again the same question. For the answer that you can go as cytokinin, because it's go with the cytokinin. Since it's having a cytokinin, where it goes with this rich source of cytokinin, there you could see in the coconut milk. These are the plant growth hormones which are basic in nature, which promotes what? Cytokinesis. You know cytokinesis, where the cytoplasm will start dividing, and also moreover you could see in coconut milk endosperm formation. The previous chapter we studied cellular endosperms and nuclear endosperm, the tender coconut, the milk and the kernel, this part that if you can recollect, right, where what happens, like, the nucleus which will be keep on dividing inside, and the nucleus may be like accumulating. Then what happens, like cytoplasm which divides, and what happens, it leads to multinucleate stage. This is a characteristic feature where the free nuclear endosperm and that is free nuclear endosperm and cellular endosperm can be formed. This because of what? Because of the cytokinin, and this promotes cytokinesis, not the karyokinesis. Getting what I am trying to say? So this which occurs, and this leads to the conjunction with the auxins in plant tissue culture experiment. Coconut milk is widely used as a nutritive medium, because when you talk about this sexual reproduction also, where the zygote will develop to embryo, it will wait for some time for the development of endosperm. After the development of endosperm only, the zygote, the embryo start developing. Reason is because endosperm is a nutritious medium, because for the zygote, the embryo which need nutrition for its further development, for that it requires what? Endosperm. It clearly tells like the coconut milk is a nutritious medium. So that is the reason why the coconut milk often used in the artificial environment, the tissue culture, for the nutrition purpose, because endosperm. Okay, because the concept is this. Maybe the question comes in different part, like they just manipulate and ask in whichever the form, and you must be in a position to answer. You need to correlate what you studied in sexual reproduction, what you studied in the plant kingdom, what you studied in the plant physiology. You need to correlate, and if you know the concept clearly, you can connect it anywhere and you can give the correct answer. Okay, just little bit of understanding only that you require to answer this type of question. Okay, we move on to the next question. Question 105. Horizontal growth of seedlings, swelling of the axis and apical hook formation and dicot seedling is brought about by, this is by means of ethylene. It's a direct question. So horizontal growth of seedling, this because of ethylene. Option 3 is the correct answer. Ripening of a fruit. Ripening of a fruit is controlled by, so I don't want the ripening of fruit, because if you exporting the fruits from one country to the other country, the ripening should not occur like within a timely fashion, because it should not be degraded or decomposed, like the vigors should be maintained properly. Then what we have to do is, we need to apply this controlling. The ripening of the fruit can be controlled by means of ethylene. So option 1 is the correct answer. Question 107. Which of the following hormone growth regulators are called as stress hormone? Stress hormone, and case of animals, mammals, we call it as epinephrine and norepinephrine. In plants, which is called as a stress hormone, it's called as ABA, that is abscisic acid. Guys, question number 107, abscisic acid, which we call it as a stress hormone, which is a stress hormone. During stress or emergency conditions, the plant also which requires this type of hormone, this we call it as abscisic acid. So 1 is the correct answer. Question number 108. Before that, we go to the next question. Why we call it this is a emergency hormone? This because of the stomatal closure, and for other reaction, even the stress conditions, the like drought or the famine conditions, or the plants which cannot grow, but this cannot die until the unfavorable condition gets over. Somehow the plant is supposed to survive. For that, which hormone is responsible for making like courage, giving a courage for the plants to grow? Because one point of time the unfavorable condition will turn to the favorable condition. It has to wait. Which hormone is giving this confidence and courage to the plants? Only the stress hormone, that is abscisic acid. Okay, and also the stomatal opening and closure, when it has to open, when it has to close, and this kind of adjustments or manipulations which can be done in the emergency situations. So by means of abscisic acid, this you need to keep it in our mind. Specifically, they give any one drought condition or famine condition, opening and closure of stomata, any one example that they'll give, and they'll ask you which hormone will be taking place, functioning in this particular situation. Means that also you should remember abscisic acid. Okay. So next question, question 108. Which of the following growth hormones is associated with the stomatal moon? So just now I told you, this type questions also will come. If they give just an example itself, function itself, then also you need to go with the abscisic acid, that is ABA, the stress hormone. Then question number 109. The light duration required for the plant to produce flowers is a critical daylight. We told you short day plants, long day plants. I hope you would have not forgotten this one. Critical day length, because light is the factor. That is why I told you short day plants, long day plants. We never call it as short night plant, long night, because Darkness is not the factor. When you switch off the light, it will become dark. So dark is not the factor. Here the light is the factor. Having light is the factor, based on that only we call it as, here the critical point is based on what? Based on the light source only. Light is the factor. So this light duration required for a plant to produce flowers, here the flowering is depend on what? On the light source. Maybe short day plant or long day plants, but here the light is the factor. So critical day length is the correct answer. I hope we discussed already in the class part, so I hope you won't be getting doubts in this question. So question 1092 is the correct answer. 1110, if a plant produces flowers when exposed only to alternating periods of 5 hours light and 3 hours dark in a 24-hour cycle, then the plant is long day cycle. It's very quite common. The concept that you should is like, this is the day part. For example, it is a dark part. For example, I'm shading this region. This is a dark region. This is a dark part. And this is a light region. You know what happens, like PR, PFR, this concept I told you already. PR will get converted to PFR, that is for phytochrome red to phytochrome far red. So what happens, phytochrome red will convert it to phytochrome far red, and phytochrome far red will be converted to phytochrome red. As far as long day plants are concerned, which is required, the light is important factor, where it requires more amount of light. But also I told in the class session, the session itself, if this light is interrupted by means of some types of darkness, again the flowering will not be induced. It means light is essential for this long day plants. You must give uninterrupted. There should not be any interruption. You switching off the light and giving some darkness for 2 or 3 minutes in between. If you expect flowering in that particular plants, no, it will not flower. Right? How it can be the long day plants, the requirement is what? The light which starts from year to year means continuously the light source has to be given. If you are stopping the light source in between, if you are keeping the plant in the dark condition, then definitely, since the light is an important factor for the long day plants, the plant will not grow because it requires uninterrupted light source, but darkness should be very short and more must be the light. The same way, the vice versa, short day plants where the light should be less and the darkness should be more. If you interrupt the darkness by means of the light, again the plant will not induce flowering. This is the basic concept for the neutral plants, but neutral plants least bother about anything. Neutral plants leave it, but long day plants and short day plants follow the principle of this basic concept. So having this in your mind, we go with the long day plant. So option 4 is the correct answer. Question 111. Day neutral plants relates to flowering in, I told you, the day neutral plants least bother about the light or day. It induces combination, flowering in all possible photo periods, it can bloom. So no issues with that. So don't think like neutral plants will glow only in the daytime and night time. It requires only this much of neutral plants are neutral in condition, irrespective of whatever the neutral condition that you're providing, like more of light or less of light, it least bother where the flowering can be induced in that type of plants. So option 3 is the correct answer for neutral plants. Question 112. Which one of the following synthetic growth regulators is used to promote synchronized flowering in pineapple? In this case, we go with option 4, that is 2-chloroethyl phosphonic acid, because 2-chloroethyl phosphonic acid is a synthetic ethylene. You know, this is a synthetic ethylene and which is commonly used for commercial purposes where it stimulates flowering. In example, I told in the pineapple. You might have not forgotten this pineapple, and helps in the synchronizing the fruit set. Sometimes the pineapple may not be having the spiky thing and it will be very short. This is because of what? So what we farmers we do is like, we increase the size of the pineapple. How? By means of applying the synthetic hormone, that is 2-chloroethyl phosphonic acid. This is quite related with the pineapples. So option 4 is the correct answer. Question 113. The phenomenon of vernalization can be seen. So in sugar beet, cabbage, carrot, this the options they given for this, all of this, because what is vernalization? Is the process of shortening a vegetative phase unnecessarily, because we not continue the life cycle. So what I'm going to do is I'm going to shorten the vegetative phase. The vegetative, because as far as the farmer is concerned, which phase is like essential? Only the reproductive phase. Unnecessarily, why I need to supply the nutrients in terms of like vegetative phase? So I'll reduce the time span of the vegetative phase. I give more importance for the reproductive phase. So what happens? It will be beneficial. So unnecessarily, for example, it's something like the childhood. As far as the human beings, we cannot do this case of human beings. I don't want like the child phase, I'll just trim off the childhood, I can go directly to the adolescent phase. I cannot do with the human beings, but the plants I can do it. What I can do is, I can chop off, I can reduce the vegetative phase, so directly I can make the plants to directly get inside the reproductive phase, where it will be beneficial for the farmers to go with the crop yield. So having this in your mind, this vernalization is a process by where I will asking the flowering by the previous cold treatment. I will be exposing the plants with the cold treatment. For example, you take with cabbage, wheat, carrot, sugar beet, winter rye, barley, pea. These are the some of examples where we go with the vernalization process. So option is all of this. Question 114. The cell attaining the maximum size in terms of wall thickening and protoplasmic modification belong to which phase? Maturation phase. Because we studied like meristematic phase, elongation phase and maturation phase. It occurs only in the maturation phase, which leads to the physiological differentiation to attain the particular shape, size, thickening. That is internal constitutions at the particular function. So option 3 is the correct answer for question number 114. Question 115. The viability of the seed is attained or tested by, that is 2,3,5 triphenyl tetrazolium chloride. So question 115, option 2 is the correct answer. This is because will make the seeds to go for germinate and will make the seed stability to respire. So option 2 is the correct answer. Question number 116. Exponential growth in plants can be expressed. As far as exponential growth in plants, how can be expressed? When we go for the exponential growth, it will be expressed like this actually. So this expressed by means of W1 is equal to WOERT. So this is how you can express the exponential growth as far as your plants are concerned. Where the exponential growth, where you could say W1 is the final size and WO is the initial size. Where R is what? R is the growth rate. R is the growth rate and T is the time of growth and E is the base of the natural algorithm. So this is depends on what? The final weight, it depends upon the initial weight and the base of the natural algorithm. So for this question, option 3 is the correct answer for question 116. Next question 117. Senescence as an active development cellular response in the growth and functioning of a flowering plants is initiated by. Senescence is what? Nearing to the death phase actually, where withering of leaves, all the metabolic functions can be seized off. This particular case where the vessels and trachea differentiation usually can be indicated, because almost the plants or the particular tissues which is going to the nearing to the dead phase where no metabolic functions can be evidenced, where the cell division followed by the scent enlargement and differentiation precede the actual development. Senescence of cell in the distal region leads to the lignification. So if you lead lignification, almost what will happen? The thylose formation. You have not forgotten about thylose formation in the tracheary element and cause the callose deposition sieve elements which occurs in the advance of abscission. Abscissions we call it as a senescence. So the final lead to the actual separation. So that's what happens. Your vessels and the tracheids, the differentiation indicates senescence. Then the older part supposed to be withered off from the plants. So for this particular question, question number 117, option 3 is the correct answer. Question 118. Dr. F. Venn noted if coleoptile tips were removed and placed in agar, this we discussed already, question 118, and this is made possible the isolation, extraction, identification of auxin. This we already discussed, that is 93. So the same explanation as if for 93, 118, the coleoptile that Venn test that we have done, right? You can take the same explanation that we discussed for question number 93. Question number 119. Phototropic curvature is a result of uneven distribution of, this again the same question, the vent and ox avena we discussed. Again the repetition of the question, guys, the same explanation that I given for 93. For this also you go with the auxins, okay. So we go for the fourth option for question 119. Question 120. Auxin is a derivative. This I told you in the class session itself. If you look into the video, you can get the answer. Auxin is a derivative of tryptophan. It's a derivative of tryptophan because it's responsible for the promotion of the scent enlargement and the maintenance of the apical dominance. So for question 120, option 2 is the correct answer.